This is an area of oak woodland quite close to Exeter. Most of the oak woodlands in Devon are old coppices. Coppicing is a very ancient management practice and was originally carried out for both wood and bark for tannins, and also small wood for making charcoal, particularly for gunpowder. What coppicing involves is cutting down the trees on a rotation, and you get sprouting from the same rootstock to form these complex boles, which increase in diameter with time. And for these sort of light wood products, rotation was over about a period of 15 to 25 years. On the other hand, some of the stems are allowed to develop up through the canopy. This is probably in the order of 70 to 80 years old, and already you're beginning to develop a certain amount of dead wood up at the top. Now, the decomposition is fairly slow up there, conditions are dry, and by the time it falls, material over a period of decades has lost about 50% of its weight. But when finally it hits the ground, it reaches an environment which is far more favourable for decomposition. The conditions are far moister, and you get very rapid fungal growth into the woody material. But finally, of course, the wood has been extensively invaded by fungal growths. The animals move in and start breaking down the material really rapidly, and it disintegrates into the soil in no time at all. As you can see, it's very, very heavily invaded with a certain amount of frass from fly larvae and millipedes and so on, and that will disappear down into the humus quite rapidly from that stage. This time of the year, in early October, the leaf litter is getting fairly sparse on the surface of the ground. As you can see, there's fairly thin leaf litter cover. In about a month or so, we'll get 300 grams of leaf litter per metre squared falling from the trees. You can see the first of the autumn leaves just beginning to come down now. Now, I suppose during the course of 18 months or so, most of this leaf litter will actually break down, but the actual total course of decay is much longer. The oak leaf litter's got about 1% or less of nitrogen, 20–25% lignin, and lots of tannins you might expect of oak, and the overall time course of decomposition is in fact fairly slow. As a consequence of this, characteristically under oaks, you get a rather acid organic soil profile. Look, I'll show you. Right, so what we've got here is about 5 centimetres of very compact organic matter with a very sharp boundary onto the mineral B horizons, the pale or inorganic material underneath. Now, the organic material here in the humus horizon is this very fine, only partially decomposed humus, which is largely composed of the feces of very small soil animals. And this is the thing which characterises what we know as a mode of humus form, sometimes called an insect moll. Now, what are the animals that are responsible, other organisms responsible for the decomposition nutrient cycling? Let's see what we can find. Well, the surface leaf litter horizons are often associated with the macroarthropods, things such as millipedes and woodlice. In fact, here we have the pill millipede, Glomerus marginata. This one rolls up into a ball. Macroine fauna include woodlice. This one is Aniscus ocellus, which is very common in acid woodlands. Also in these surface fragmented leaf litter horizons, we've got certain acid-tolerant earthworms. This one is Lumbricus rubellus, which is typical of these earthworms living in the surface horizons. And these are really important in shredding the leaf litter and working it down into the surface of the soil. So when I say litter disappears in about 18 months in this site, we're really talking about the role of these animals in fragmenting it, and also the physical processes such as wind blow and so on. Of course, all these groups have predatory members. One of the ones which I particularly fond of are the pseudoscorpions. Pseudoscorpion because they don't have a sting, but they are in fact voracious predators, to particularly attack the collembola in a very spectacular way. Very small collembola, such as these white ones, Folsomia, which characteristically live in the surface horizons. They're about 1 millimetre or so long. They also live in the compact areas further down. And enormous numbers of mites, which live particularly in the fermentation layer between the surface litter and the underlying humus. Now, the role of these animals is principally breaking down the feces from macroarthropods and other animals and leaf material into very, very fine particulate frass. Also included in the mesofauna are the encotrade worms, tiny white worms, sometimes as small as a few millimetres in length. Others grow up to a couple of centimetres long. You don't see an awful lot of them, but they are in fact very important in reworking the fecal material and frass from other soil animals.
Now, as we go below this fermentation layer, we come into the beginnings of the root mat, and here we can see very characteristically the mycorrhizae associated with the oak roots. Now, the mycorrhizae are a symbiotic association with a number of different basidiomycete fungi, and it's a very effective mechanism for picking up nutrients. Now, if you imagine at this time of the year when the leaves are falling and you're getting a certain amount of decomposition nutrient release, the plants aren't actually taking up the nitrogen, phosphorus as well. And the role of mycorrhizae is in sequestering these, storing it within the mycorrhizal network, and handing it out to the tree next spring. Now, as you go down below this root layer, you begin to get into the compact humus horizons. Now, this whole area where the roots are active, and at the beginning of this profile, is an area of intense biological activity. Here we've got hundreds, thousands of microarthropods, collembola of mites. In fact, in a well-developed woodland soil like this, there could be 300,000 of these tiny animals per metre squared. But even more remarkable are the ones that you don't tend to see. There could be 10 or 20 million nematodes per metre squared, and in a tiny amount of soil organic matter, in its wet state no more than the size of a sugar lump, but in its dry state about a centimetre cubed and weighing about 1 gram, we've calculated that for this particular site, there are between 10 million and 100 million bacteria and somewhere between 3 and 30 kilometres of fungal hyphae per gram of soil. Now, the big question is, what do all these organisms do? As I say, the macroarthropods are chewing up the leaf litter, the microarthropods and other small soil animals further breaking it down into this fine material. The protozoa and the nematodes, which we can't really see, but they are feeding on the bacteria, grazing on the fungal hyphae, rather like cows grazing on a lawn and releasing the nutrients that way. But how do they really interact? Well, that's an incredibly difficult question to ask out here in the field. What we really have to do is go back into the laboratory and look at it under rather more defined, prescribed experimental conditions. This mass of microarthropods has been extracted from only a handful of litter. But although there are millions of these animals per metre squared, their contribution to the carbon metabolism is only about 5–10%. 80 or 90% of all the mineralization of the organic matter entering the soil is by fungi and bacteria. So what are the animals doing? Well, there's a number of ideas that have been put forward for years. One, that they spread the spores around, and two, that through breaking down the leaf litter into these fine particles, that they enhance fungal and bacterial activity. Well, one of our first objectives is to see whether this actually does occur. So what we did was to grind the litter down to very small defined particle sizes and deposit them onto filters. These are then inoculated with fungi as a basidiomycete, which will decompose both lignin and cellulose. And then these are put into a respirometer. And here we measure fungal respiration. What we've got is a small container containing potassium hydroxide. And as CO2 is released by the fungus, it's absorbed in this KOH, and this produces a pressure reduction, which is then measured by this manometer.
And by adjusting the vernier, we can see that pressure change and get an estimate of fungal respiration. So this was carried out with a variety of different particle sizes over a number of weeks, and these are some of the results here. And what we can see is the respiration of 3 different sizes of particles. So we have time in weeks and the fungal respiration. And what we can see is that the large pellets show a peak of respiration which dies away with time, and this increases through the medium pellets through to the smallest size range, which show a tremendous enhancement of fungal respiration. But in all 3 cases, this dies away with time, and what this shows is that through this comminution, through the fragmentation of the litter, we're mobilizing some labile pool of litter carbon. Now, in the next experiments, we use exactly the same technique, but we concentrated the litter from the initial sort of filter that you saw into, first of all, half the surface area filter, and then a quarter of the surface area filter. So we're producing the sort of concentration, the aggregation of particles that you might either get in feces or in the humus layer. We're now plotting the mean particle size against fungal respiration. In the smallest particles, we get this enhanced respiration, and as the particles get larger, the microbial respiration declines. But if we've concentrated this material down into these sort of matted aggregates, we find as the particle size declines, so does microbial respiration. Perhaps this is one of the reasons why in these monohumus soils we're getting this accumulation of organic material. Well, what happens if we put the animals in? This is what you might imagine happens to fecal material after it's voided from the animal. Let's look at a more dynamic situation with animal feeding activities in situ. Got a large number of experimental chambers stored in these cabinets at constant temperature. Now in here we've got leaf litter, oak leaf litter, which has been broken down to a series of coarse fragments, and in here we put different numbers of wood lice or millipedes. And the carbon dioxide released from the animal microbial respiration is absorbed in a little dish attached to the lid, and we determine the total amount of CO2 absorbed by titration with alkali. Now the results of a series of experiments show this sort of pattern, that if we plot the number of animals, including controls without animals, against the total amount of microbial respiration over a period of weeks, and what we can see in the top trace is that as the number of animals increase, it reaches a maximum, and then as the number of animals continue to increase in the experimental chambers, microbial respiration declines. If we compare that with the respiration in chambers which are prepared with material ground down to about the size of particles you find in millipede or woodlice feces, this enhancement and subsequent decline is far less marked. An interpretation of this decline in microbial respiration, which occurs in experimental chambers with microarthropods as well as these millipedes and woodlice, is that you're getting a reduction in fungal biomass and an increase in bacteria, largely a result of bacteria increasing as the material passes through the animal guts. The fungal thallus is very sensitive to disruption, and less so in the case of the single unicellular thallus of the bacteria. Now, immediately we discovered this phenomenon, we wondered what the consequences of this is for nitrogen mineralization, which involves both fungal immobilization of nitrogen and also the bacterially mediated steps such as nitrification. And to investigate this, we basically used the same type of experimental chamber, but this time we flushed through the leaf litter with distilled water and then analyzed the leachates for nitrate and ammonium using an autoanalyzer. Samples of leachates are pumped out and mixed with reagents, and then these pass through a series of mixing columns to allow color development. The intensity of color is measured in a colorimeter, and then the concentration of nutrients, which are proportional to color development, is expressed as a series of curves on the chart recorder.
leaching and analysis of the samples is carried out weekly, and this is a typical pattern of the results. The bottom trace is nitrogen mineralization by microorganisms in the absence of animals, and here we can see over a course of about 20 weeks, and about 3% of the total nitrogen in the leaf litter in the experimental chambers was released. In contrast to the treatments with animals, where you get a massive early flush of nitrogen mineralization, and this represents about 15% of the mineral nitrogen in the leaf litter. So again, we can see that the animals are releasing some labile pool of nitrogen, and this is occurring at a far earlier stage than it would occur with microorganisms alone. The time course of nitrogen mineralization simply with bacterial and fungal processes might take 2 or 3 times the length of this experimental period. In addition, we removed the animals from some treatments, such as this trace here, and what we found is that it takes time for the effects of the animals to die away. Now, it's all very well showing these effects in the laboratory, but how relevant are they to the field situation? We've got to first of all demonstrate that they quantitatively occur and that they're ecologically significant at an ecosystem level. This is an experiment set up at the end of 1981 to look at the effects of soil animals on nitrogen mineralization. What we've got in this area is leaf litter traps to measure the amount of leaves falling in the autumn. These rainfall collectors to measure the amount of nutrients falling out of the canopy, and of course temperature, soil and air temperature measurements. Now, the details of the experimental setup are this. These lysimeters are 1/4 metre square and about 30 centimetres deep, and they've got a mesh and bead drainage bed in the bottom so that the rainfall falling in drains away into these collectors which are down this trench, and then we can compare the concentrations of nutrients in these solutions with those of the incoming rainfall. Now we've got a number of different treatments. The ones along the outside are with roots, tree roots let in through ports on the side, and then we've got individual chambers, some with leaf litter and animals, and some just with leaf litter alone. So we've got 4 treatments, with or without roots and with or without animals. Now, if we look at this one here, this is one of the treatments without roots and animals. What we've basically done is to set it up with soil organic matter and leaf litter at approximately the amounts and falling rates for the site. And we've put over the top this mesh to keep out the macrofauna. And this leaf litter is now about a year old, and you can see that most of the material is there. It hasn't really broken down at all. Now, if we compare that with the treatment over here, which has had animals in, and they have extensively broken down the leaf litter. Open it. And here you can see that after the same period of time as you saw in the other treatment, the leaf litter has been very extensively broken down. It's still here, but a lot of the leaves are reduced to skeletons and fragments. And in fact, the whole bottom of the chamber looks completely different because it's a mass of frass and so on. What we've assumed, of course, is that the collembola, the mites and the nematodes and so on are the same in all treatments. It's very difficult with this sort of experimental system to control those as well. So what does this mean in terms of the actual nitrogen mineralization? Well, what I've done is to bring some computer printout here with some results. What we've got here is the ammonium concentration which is coming out into those bottles, the season in weeks here with the summer period and the autumn and the following spring. And the main patterns to note are that here in red we have the treatments with the macrofauna. Underneath is the treatments without macrofauna, and here the treatments with roots. And basically what you can see is that the macrofauna are the highest trace all the way through the summer. This represents the additional amount of nitrogen mobilized from these chambers by the activities of the animals. But in winter, curiously enough, it swaps over. And we're not quite sure why this is, that in fact during the winter period, less nitrogen is lost from the lysimeters with animals than those without. In the presence of roots, the roots take up the nitrogen from the soil as it's mobilized. And again, we have a curious enhancement effect that the treatments with roots and animals, this bottom trace, the actual amount of nitrogen which is lost is lower from the treatments with roots alone. But not all the nitrogen is mobilized as ammonium. In acid soils in general, you don't tend to get much nitrification. This partly reflects the efficiency of the roots in taking it up, as well as the acidity of the soil, which tends to inhibit the activity of nitrifying bacteria. If we look at these results here, where we've got cumulative amounts of nitrate from the lysimeters with time, you can see the amounts coming out of the rooted treatments are very low. Interestingly enough, we get this consistent pattern that in the treatments with roots and animals, we appear to have a more effective uptake of nitrogen. But if we look in these two traces here, we get very high amounts of nitrate coming out because we haven't got the roots to take away that ammonium. So that in the treatments without animals, and particularly in the ones with animals, we've got high degree of nitrification. This is unexpected because it's often assumed that the tannins in oak leaf litter will actually inhibit nitrifying activity. In the second year, we put two additional treatments into the system. One, we applied pesticides to some of the treatments with animals, and the others, we cut some of the roots. What this showed is that when we applied the pesticides and killed off all the millipedes and woodlice and so on, in fact, the nitrogen losses from the lysimeters didn't change significantly. This reflects the fact that the animals had to build up these conditions in the soil through interactions with microorganisms over a period of weeks or even years to form what are now completely different soil systems in each one of these lysimeters. In the case of the roots, when we cut the roots, predictably, within a few weeks, we've got a tremendous loss of nitrate from the chambers. This is very similar to the sort of consequences of clear felling, where you disrupt the soil-root link, remove the tree uptake of ammonium, free the ammonium for uptake by nitrifiers, and very characteristically get large losses of nitrogen from the system. So overall, what these experiments have shown is that very small amounts of macrofauna have very large effects, and we've now been able to interpret these in terms of kilograms per hectare at an ecosystem level.